you, Athena, for organizing this event. I'm really excited to present some of the past work that I have been doing around insects and their microbiome. And I decided to entitle this presentation as The Secret Superpowers of Insects and Other Creatures. As Athena mentioned, I am a scientist at the Berkeley lab, and that's my contact information. So, all right, there you saw my whole screen, and now you see that. So, okay, we know that arthropods and insects are pretty much everywhere, right? I mean, we can find them in soil, in water, in plants. Some arthropods are able even to live together with animals, even in the hair of humans, and they are just impressive creatures. The thing is that when most of us think about insects, we immediately go to a scenario like this, just thinking about what can cause diseases or insects that can be pesky. However, insects are impressive creatures. They appeared on our planet like around 300 million years ago. And during this time, well, they learned how to associate with plants and bacteria. You know, they have had a lot of time to interact with these other groups of organisms. And through this, they have created impressive associations with microbes, again, with plants. So to me, if I think about insects, I see them as creatures that are empowered by microbes. I will ask something right now. Can you still hear me and see my presentation?
Yes.
Yes, with you.
Awesome. Okay. So, okay, I will get here. So to me, insects are something like this, super creatures. And in the laboratory and through my time at LBNL, I have been working with what I consider amazing organisms, non-model insects. And one of them is this tiny little insect called the coffee berry borer. This is Hypothenemus hampe. This is an insect that is like 1 millimeter size and is a very important pest because it's able to feed in coffee. And while doing this, the insect is actually exposing itself to toxic levels of caffeine. So if we try to quantify the levels of caffeine that this organism is exposing itself to, in one day, it would be the equivalent to the caffeine containing 500 shots of espresso. So to me, that just makes him a super insect. So what we did is that we went to collect samples around the world and characterize a microbiome, and we saw that all these insects from different coffee-producing countries had like what we can call a core microbiome. So what we wanted to know were whether these microbes were able to actually degrade the caffeine. So we dissected the beetles from different countries, and from there, we used agar plates at the beginning. These plates contain caffeine as the sole source of carbon and nitrogen. There was nothing else but the agar and some salts. And there you can see that we prepare a gradient of caffeine from low to high, and you can see microbes growing in the caffeine. To further test whether they were able to grow in pure caffeine, well, we transferred them to liquid media where you don't have the carbon from the agar. And actually, all of them were able to grow. And these are the different groups of bacteria we identified. As you can see, we have a quite large number of bacteria that are able to survive, again, in pure caffeine. So the question was that now, are these microbes helping the beetle to transform the caffeine? I mean, you know, you can have the trait of the organism, but maybe not active within the host. So we did some RNA analysis, and we actually discovered that most of these organisms were active. At least genes involved in caffeine degradation were active within the beetle. And then, well, we set up this experiment that I'm showing here. We have the coffee beetle here, and we transferred it to regular diet. We left it there in the diet for 4 weeks. Then from there, it was transferred to this other set of diet, which is coffee beans and agar, with fluorospheres that you will see here. So then we waited for the beetles to produce some of their frass, and then we measured the caffeine in the frass in these tiny amounts of caffeine. And then we had the same, but now we treated the beetles with antibiotics, and then they went to the fluorospheres. And what we observe here is that those beetles that had their microbiome intact were releasing no caffeine in the frass, while those treated with antibiotics release levels of caffeine that were really similar to the caffeine in the diet, the initial diet. So then a next experiment that we did is that, again, we treat a population of insects with antibiotics, but then we inoculated them with one isolate only out of the several that I showed you in the previous slide. And then again, they went to fluorospheres, and we observed that actually the beetle was again, or the microbes were transforming the caffeine within the beetle. And with this, well, that we observed that the coffee berry border relies on its microbiome to transform caffeine and survive the ingestion of lethal doses of the alkaloid. Again, this is really like for me, it's like a commercial of the work that I'm doing. So we can talk a lot more like in a separate conversation about these experiments if anybody's interested. But what I'm going to do here is I'm going to jump through some quick topics that I have going on, because we only have 7 minutes and I already used one, I think, just telling you this. So the other system that we study in my laboratory is the passalid beetle. Passalids are interesting organisms because they are subsocial. They communicate with each other with sound and with chemical signals. And beyond all that, they are wood-degrading machines. They actually can consume around, one insect can consume around 1 kilogram of wood in less than 1 year. So because of this, we got the interest to understand how the beetle manages to survive in only wood, and what are the type of processes that are actually mediating the transformation of the lignocellulose. So here is an image, a picture that I made for the digestive system of the beetle. This is how you see it arranged within the animal. And well, I didn't mention this. This is a disclaimer. I'm a chemical engineer, so I started to think about this beetle as a set of bioreactors. And again, as a chemical engineer, I wanted to understand what were the environmental conditions within the beetle gut that may be important to mediate the transformation of the lignocellulose. So what I did is that I dissected the beetle. I didn't digest any insects, I promise, but that I dissected the beetle, and then I used microelectrodes, as I am showing here, to measure the gradients of oxygen in the different parts of the beetle's digestive system. And this is something that I'm illustrating here. You can see the same colors as I have in the main diagram. We have the foregut, midgut, anterior, and posterior hindgut. And where you have more blue color is where we detected more oxygen. And then towards the center, in all cases, we had an anaerobic core, which gave us the idea, well, we must have the co-occurrence of aerobic and anaerobic processes. We did a lot of chemical analysis to understand more what was happening in each of these regions of the beetle gut. And then we went to do metagenomics and also transcriptomics. And this very busy image that I'm showing you here is showing, for example, that we detected genes coding for enzymes that are involved in the transformation in lignin in the 4 regions of the beetle gut, as well as different glycoside hydrolases that may be important for the transformation of cellulose and hemicellulose. The important part here is that we detected that these enzymes were higher in the midgut and in the posterior hindgut, while lower in the anterior hindgut, where we detected that it was pretty much an anaerobic environment. We detected also the proteins using proteomics involved in these processes, and they pretty much follow the same patterns as we detected with the metagenomics, meaning you have more lignin-degrading enzymes in the midgut and posterior hindgut, and well, glycoside hydrolysis were pretty much distributed along the beetle. So using genome binning and analyzing further our metagenomics data, we were able to differentiate the processes that occur in every part of the beetle gut, as well as identifying the different organisms that contribute to the transformation of the lignocellulose and the beetle gut. We also did an analysis of metabolomics in the frass, and we compared it to that of the wood, and we observed that actually the frass is released as an enriched substrate that contains a lot of metabolites that are known to be important for insect development. And from this, we can pretty much explain the fact that usually babies or larvae spend the whole day following their parents, just waiting for these droplets to come out so they can actually get some nutrients. Because let's consider that these insects have a non-well-developed mandible, sorry, the larvae only. So they need to actually feed on the frass that is produced by the adults, so they can actually get some nutrients out of living in that wood. So this has changed really how we think about insects in general. And now I'm working in exploring the microbiome of insects in soil ecosystems. So here I'm targeting all types of insects and arthropods and characterizing the microbiome while also isolating some of these microorganisms that are able, for example, to degrade lignin, cellulose, hemicellulose, or even fixed nitrogen. And well, the ideas and possibilities are quite big after this experiment. So I'm going to close my presentation now. Again, I'm Javier Ceja-Navarro from the Berkeley Lab, and there is my website and my Twitter handle, and please feel free to contact me anytime. Thank you.
Thank you for the great talk. Questions for Javier. Cynthia?
Thanks for an exciting talk, Javier. I wonder if in the first part of your talk, the microbes that live in the gut of the coffee, are they able to live in other environments that are not necessarily the gut of this insect, like for example mammals?
For example, what? Sorry, mammals?
Yeah, mammals.
You know, I mean, if you saw the names that I presented there, I mean, we can find them in for example, soil overall. Some of these organisms, many of them were enterobacteria, so I'm sure that you could find some of these also in other organisms and other insects. I cannot tell you really if it's in mammals, but it would be interesting just to know if they are. Again, you know, you can have. There is one thing that I need to mention. Some of these organisms are also fine. You can also find them in soil, for example, but that doesn't mean that they are transforming the caffeine. Maybe they are just doing some other things in there while having the genetic potential. Maybe in the gut, you have the pressure from the environment, the type of ecosystem that you have, so they express the caffeine degradation. And I went far from this. Yeah. Thank you.
I see some comments about your amazing slides. Javier actually drew most of those, if you weren't impressive enough with his science. One more question from, sorry, I'm pronouncing your name wrong probably. Jigyasa?
Hello, yes, you pronounced it perfectly. This was an excellent presentation. I work with termites, so another wood-feeding insect.
Yes.
I was wondering, you talked about caffeine digestion. Have you found like genes specific for that? Yeah, go ahead.
Yes, because we isolated the organisms and we also characterize the genomes. So we have the information and the different operons that are involved in caffeine degradation, which are not the same in the different organisms. That's something that I should say that I found super interesting. In Pseudomonas, for example, Pseudomonas putide and Pseudomonas fulvae, there is an operon for that includes caffeine demethylases, but in the other organisms you have different strategies for caffeine degradation. So yes, we have studied that, and it's super interesting thing that I should work on and publish maybe. But yes.
Oh, that's great. Is it possible that I ask another question, or maybe I can contact you later if there's not time?
Yeah, go for it.
Okay. You mentioned lignin genes. Have you tested if those peroxidases that digest lignin are actually active, or they're just present in the metagenomes? Because this is one major thing that we find that in termites too, and you don't know if they're active.
Okay, one thing that we did is that we use a chemical analysis called thermochemolysis, and I'm sure that you have heard of it. So we use that one to actually elucidate the mechanisms for lignin degradation in the beetle. Then after, we knew that it was happening, and we know that there are chemical modifications of lignin occurring, right? When you compare the wood with the products in the frass. We also, from some of these genes, we did qPCR in RNA that was extracted from the beetles, and we see at least that we can detect levels of expression of them. That's the extent that we have done so far. And we also have prepared, for example, phosphate libraries, and we can see expression of some of these genes when in our phosphate. Yeah.
All right, thank you. That's great. You've done a lot of work here.
Great. And before moving on, I want to do a shameless plug for a project that me and Javier have been working on since like the end of 2019. We're actually organizing a microbiome workshop and it will have a focus on host-associated system and non-model organism. It's going to be a 2-day event. The first will be a symposium, which is just like a more three-dimensional version of this. And then the second day will be a hands-on workshop. And we're very happy to have gotten sponsorship from Oxford Nanopore and NEB. So we'll be exploring long-read sequencing and microbiome. This was planned for September this year at the Cal Academy. The date is kind of up in the air right now, but it will happen and we'll keep you posted. All right. Thank you, Javier. Our second speaker is Dr. Panpim Thongsripong. She is a postdoc at the Cal Academy, and she's going to tell us about mosquito microbiomes.
All right. Okay, let me do this little magic here. Share the screen. And, okay. Can everybody see the slide?
Yeah.
Okay. All right, good morning, everybody, and thanks, Athena, for arranging this awesome series of talks. So today, the start of my talk is something that everybody probably have been thinking about and reading about and talking about in the past weeks. And we're not going to be talking about coronavirus today, but we're going to be talking about mosquito-associated viromes. And honestly, I think everybody should be thinking about viruses because they are the most abundant biological entities on Earth. And according to some estimates, there are over 10 to the 30th of a million viruses on Earth. They infected almost all life forms, including bacteria, archaea, and eukaryotes, and there are even viruses of virus. And we have heard of virus because of all the bad things that they do. We know that they cause disease in humans, but they can also threaten our economy, our food sources, and also our animals. But what we don't talk a lot is the good things that they do. And in fact, virus is a very important player in the ecosystems. According to one study that looking at marine system, they estimated roughly 100 million metric tons of microbes die every minute due to viruses, specifically the phages, which infect bacteria. So if we take the virus out of the equation, then the ecosystem is definitely going to collapse. And so that's why there's a lot of people study viruses. And in the age of next generation sequencing, we see a jump in the newly discovered virus species. And this is a picture that was taken from a study that was published in 2016 in Nature, and it was one of the big study. And the authors pretty much just pull a bunch of invertebrates together and sequence everything. And so they saw tons and tons of new viruses, which are represented by the red branch shown in the pictures. So you can see that they discover new, even like an entirely new clade of viruses in this study. And even though we see a lot of new viruses, a lot of the basic questions are still unanswered. For example, we don't really understand entirely what determines the virus community composition and diversities. And because the virus are tightly linked to the host, they achieve their function only after entering into the host. Virus diversification probably depends upon a selection of host, the mode of transmission, and the strategy of replication within a cell. But what about the habitat of the host? How does the habitat of the host play a role in determining this interaction between host and virus? So to getting at that, we're going to be looking at mosquito viromes. And so I work in Dr. Shannon Bennett's lab, and we study mosquito-borne disease transmissions such as dengue virus and Zika virus. And this picture just kind of summarizes the environment that one mosquito may, live in and experience. And so the mosquito go through this aquatic life stage early on, so they may expose to different environment in this aquatic environment. And then when they become an adult, the mosquito will go around and bite, different animals and humans, and may acquire virus and transmit viruses to the host, to the blood host. And so the question is, how does this different habitats that this, for example, one species the mosquito lived in, play a role in determining the mosquito microbiome and viromes and eventually affect disease transmission? But today, the really short talk here is just going to focus on this one question, which is mosquito virome composition determined primarily by the host species, in this case it's the mosquito species, or by the habitat type of the mosquitoes? And so to answer this specific question, we collected mosquitoes in Thailand from 3 different habitat types, from rural, which the picture is shown here in the upper pictures, and then suburban, which is this one, SU, and then UR represent urban environment in Nakhon Nayok town in central Thailand. And this field work was done in 2008. So it was a while ago, and I was surprised that the RNA lasted long enough in the freezer for us to sequence anything, but it worked. So in each of these habitat type, we collect 3 mosquito species, and actually they're in different genera, and they're quite different in terms of biology and ecology. We have Amidris mosquitoes, we have Culex mosquito, and the Mansonia mosquitoes. And so in total, we have 9 samples, 3 mosquito species and 3 habitats. And in each sample, we put 25 mosquito of the same species from the same habitat together, and then extracted the total RNA. And we know that a lot of these RNA is going to be mosquito ribosomal RNA, but we want to sequence this tiny amount of microbial RNA. So we actually remove the host ribosomal RNA in the lab before we sequence the shotgun sequencing the cDNA libraries. And the removal of the ribosomal RNA, we use the lab-made probe. And so quickly to the result here, we actually have 2 sets of samples. The first set is here, RD sample represent the samples that went through this ribosomal depletion steps. And the UD samples are kind of like a control sample where we didn't, the sample didn't go through this ribosomal depletion step, so it was undepleted. And we see that we have more viruses. We found more viruses in the RD sample. So the samples that the ribosomal DNA was depleted, we found more viruses, we found more virus contig. And we also see higher average percentage of virus reads, about 0.4% of virus reads compared to the 0.09 in the samples that the control samples. We also have a higher depth of the virus in the RD samples as well. And so when we classify the virus reads, this show the virus family distribution of the showing the family of virus closely related to the virus reads in our samples. So the species of mosquitoes are shown up here, the Amidris, the Culex, and the Mansonia, and these are the negative control. And the samples are shown in the habitat types are shown in the bottom. RD represent the samples that went through ribosomal depletion steps, and the UD are the one that did go through that step. And we started to see some pattern here. But before we go into patterns, I'm just gonna talk about one example of some of the virus that we have seen. I'm not gonna go through everything, so just a quick sample here. So this is a phylogenetic tree showing viruses that we found in the family Nanaviridae and a closely related group of Nanaviridae. So Nanaviridae is classically classified as a fungal virus and a yeast virus. So there's 2 genera in that family. The mitovirus is considered a fungal virus, and the nanavirus is considered a yeast virus. There's also a floating genus here up here called Wormia virus that is considered a plant virus. So we've got one virus in our sample that we named the Nakhon Wormia-like virus up here. And this virus was found in all Amidris samples, but not in other species of mosquitoes. And it falls within this newly classified clade of virus that so far has been found only in invertebrates. And because it has been found in high number and in all amateur sample, we think that this virus is associated with mosquitoes. So mosquito is the likely host of this specific virus. In contrast, this mitovirus, for example, that we found in our sample, have been found in relatively low number and only in a sporadic number of Kulik mosquitoes. So we think this actually, and it's fall within a genus of mitovirus. So we think these viruses are actually a virus of fungi that happen to be in the mosquito. So it's actually, the mosquito might not be the host of this virus. So it's just show you an example of this level of interaction that we may have in our sample. We also characterize and classify other non-virus microbes. I'm not going to go into details here. We see bacteria, we see other kind of parasites such as trematodes, just to show you that even though it's an RNA-seq data, we can actually glean a lot of information out from this data set. And so the final slide here, when we use PCA, principal component analysis, to analyze the pattern of virus and bacteria presence and absence across the mosquito species and habitats, we see that, so to the left is the bacteria and to the right is the virus component. And we see that the virus communities were grouped based on the host species, which is represented by the color of the dots, rather than the habitat of the study, which is represented by the shape of the dots. And that is, we don't see that apparent grouping for the bacteria community. So to conclude, the mosquito-associated virome component was determined primarily by the host species rather than by the geographical locations or habitats of the mosquitoes. And ribosomal RNA depletion may help improve the sensitivity in detecting the low abundance viral sequences. And it is important to characterize non-viral microbial community associated with the virome because it may help identify the potential host of the virus. And lastly, virus is an important component of the ecosystem and very interesting to study. So I hope everybody start thinking more about virus, not necessarily coronavirus. And with that, thank you very much. I want to acknowledge a few names here, especially Shannon, and also Angus, who is Shannon's previous postdoc, and he did a lot of lab work here, and I inherit a lot of the protocol. So with that, I'm gonna, I guess, stop sharing.
Thank you, Panpim. That was great.
Thank
I don't know
you.
if you convinced us that virus is good, but sure. So Rebecca has a question. Can you repeat what you use for RNA depletion? The deplete
Yes.
RNA, mosquito
Yes.
RNA, sorry.
Yeah, so we actually made an RNA probe from mosquito that we have in the lab in Shannon's office. Actually, it's just, we just extracted DNA and then reverse transcribe the DNA into RNA and use that as a probe to pull out any mosquito RNA.
Okay. Dylan, you have a question? You can unmute.
Yes, thank you. Thank you very much. Did you find any corona or in particular beta coronaviruses? I noticed that Hubei was sister in your tree there.
No, I did not see any corona viridae samples in our mosquitoes.
Okay, great. Thank you, Panpim.
Thank you.
Okay, the next speaker is my good friend and former postdoc advisor, Dr. Michelle Trautwein. She's a curator in the entomology department at the Cal Academy, and she's going to talk about one of my favorite things, face mites. And if you don't know what that is, your life's about to be changed forever. So.
No pressure. Well, I'd like to first acknowledge Athena. Thank you for having me, but also Athena is my primary collaborator and partner in crime right now with all things face mites. So so some of this mess is her fault too. Can you guys see now?
Yes, it looks perfect.
Okay, great. Okay, so I study human face mites, and so I'm touching on the microbiome in a couple different ways. These are microscopic animals that actually live on our bodies, so I think they may count as part of our microbiome. And I'm also going to just touch a little bit on the microbiome of the microbes on us. So these are human face mites in the genus Demodex. This is a genus of mites that live in the skin of virtually all mammals. We have 2 species that live on us. You can see one of them here, very beautiful mite. Here are the 2 species. The long skinny one is Demodex folliculorum. The short and fat one is Demodex brevis. They were thought to be the same species until the 1970s, and then we found out they were 2 very distinct species, maybe millions and millions of years divergent from each other. They live in separate microhabitats in our skin, and generally we know just very, very little about these mites. So clinically, they are on virtually all of us, and they seem to be benign on almost everybody, but they are associated with a couple disorders. One, rosacea, which is a skin disease, and also blepharitis, which is inflammation of the eyelid margin. And so basically they occur in high density and high population numbers in both those conditions, but it's not known if it's causative or if it's just associated with those disorders. And so I've been interested in the global genetic diversity of these 2 species for the last few years. And what we've discovered about them is that they aren't transmitted casually. So people's face mites often have very unique haplotypes that aren't shared between individuals. So they are passed down vertically, which is from parent to offspring. And because of that, because they're not really shared casually, and they are passed down vertically, they are kind of like a proxy for your DNA in different ways. And what we found is that these mites actually have really deep mitochondrial lineages that are associated with the geographic ancestry of the host. And so what that means is that different people from different parts of the world host different face mite lineages. And these lineages are persistent. So for example, African Americans, even if they've lived in the United States for many generations, often still host distinctly African face mites. So that's really interesting. And you can see just from this picture that both brevis and folliculorum have a similar pattern, you know, as if they've gone through this kind of parallel evolution in terms of these deep mitochondrial lineages associated with geographic ancestry of the host. So in the past few years, we've sampled over 2,000 people from over 90 countries, mostly from the public floor of the Cal Academy. But in some situations, we've been able to develop collaborations and have had samples sent to us from regions of origin, from, for example, Papua New Guinea and Guam, and recently from a hunter-gatherer group in Namibia. And you can see that picture of that sampling taking place here. And so now, the last analysis that I showed you was pretty neat. What we've learned since then, that it's a much messier, much bigger picture. And so there's not this real tight one-to-one association with geographic ancestry and mite lineage, but there is this kind of geographic centering, which is now I see is very similar to human mitochondrial haplogroups. So again, it's not a super tight one-to-one, but there is this this kind of basic correlation, and it actually makes sense that this pattern would match what we know about from human mitochondrial haplogroups, because most likely mites are transmitted initially, probably in breastfeeding. They occur in nipple tissue, so traditionally it probably has been a mother to offspring transmission. So now, to understand more about this system, we've been trying to sequence a genome to understand, for example, the genetic differences between brevis and folliculorum, that long skinny mite and that short fat mite, and then also between these lineages. So how do, for example, the African lineages of both these mite species differ from the Asian lineages and from the European lineages, and so on and so on. But sequencing the genomes of these mites has proved to be really difficult. You know, it seems like now in this modern era compared to when, you know, I was getting my PhD, you just sequence a genome, you know, everyone does it, no problem. But now in terms of dealing with this microscopic multicellular eukaryote, it actually has been a huge challenge for us to sequence this genome. And so, for example, in the past few years, a tardigrade genome has been sequenced. And for those of you who aren't familiar with tardigrades, they're practically an arthropod-like thing, also microscopic, quite a bit bigger than a face mite. And to sequence that tardigrade genome, they had to use about 1 million specimens that they got from culture. So face mites can't be cultured. They can't really live off your face for very long. You know, we can keep them alive for a couple days. Maybe someone's kept them alive for a week, but the point is they're challenging to sample. So in terms of our strategies, we've sequenced individual mites. We've sequenced pooled mites, anywhere from 3 mites to 300 mites, which takes quite a long time to sample. We've tried, prior to extraction, crushing these mites. We've put them through freeze-thaw cycles. We have extended their time in proteinase K. We have, Athena uses a Kaijin micro kit to extract DNA. In terms of sequencing strategy, we've also tried whole genome amplification methods. We've sequenced on MiSeq, HiSeq, and PacBio platforms. We've sequenced complete genomic DNA from swabs. So we've just tried so many different things. And what we've found is that we can very easily amplify a ton of human DNA and a ton of bacterial DNA, and very little mite DNA. And the mite DNA that we do get is often primarily mitochondrial. So right now, our next approach is a collaboration with the Aiden Lab at Baylor College of Medicine, and a PhD student named Bryant St. Hilaire is working on sequencing a chromosome level genome based on a pooled sample that we sent. So we haven't heard yet if they've had great success, but we're keeping our fingers crossed. And so considering we get back all this bacterial data from our sequencing runs, it gives us an opportunity to look at that bycatch and get a glimpse of the face mite microbiome, which is an interesting question to me because I mentioned that these mites are benign on most people, but they do become pathogenic at some point. And so it may be that their microbiome contributes to that pathogenicity. So the bacteria that they harbor, that they spread around our face could play a role in the trouble they cause in rosacea, for example, or blepharitis. Now, the challenge in looking at the face mite microbiome is that they're immersed in human skin. So, and that's externally and internally. When you get them, in fact, you can see from this picture here, the bits they're covered with is human skin cells. They also likely consume human skin cells. They likely consume bacteria. So they really are just in the medium of human skin. So just very quickly, one of the things we've done is just scanned our data to look for, our sequencing data to look for arthropod-specific bacteria. And we have found a few examples of interesting characters that we're going to look into, including 2 different strains of Wolbachia. Previously, a paper was published specifically about looking for Wolbachia in face mites, and they didn't find any. But we have found two examples of distinct Wolbachia, so we're excited about looking into that. And for those of you who don't know, Wolbachia is a kind of well-known endosymbiont of arthropods, and it can do all kinds of crazy things and affect the reproductive strategy and fitness and all kinds of stuff of arthropods. So it'd be interesting to consider how it affects face mite diversity. So I think I'll end there, but you can see there's plenty of more face mite questions and to answer and problems to be overcome. And quickly, here is some of the large team of people that have helped me do some of this work. So.
Thanks, Michelle. Okay. Megan wants to know, can they be passed between partners or other people you touch base with?
Yeah, they can, yes. But you really, it seems like they're just people that you end up really sharing a lot of skin time with. So yes, between partners and within families.
Cynthia?
Hi, thanks, Michelle. So I wonder if the mites have different characteristics depending on the tissue they are found. That is, like if it's in the face, or in the stomach, or in the legs, because bacteria have different populations in the skin. So is it the same with mites?
Yes, I imagine that it is, and we've done, there's been one study that suggested populations might be genetically distinct in, for example, your eyelashes versus your forehead. And beyond that, I've looked just to see if, and this is not talking about the bacteria, but just the genetic, you know, the actual sort of lineage or perhaps subspecies of my, you know, is the same on myself in my ear and on my nose. But, you know, at least to me, it seems like the ear and with, you know, earwax and stuff, that's a really different kind of environment. But so that's some of the only data I have that looks at that. But yeah, you imagine if they're in your genitals or these other places, that's a long way for mites to walk on a body. Anyway, so you'd imagine they might be really quite distinct in different locations in your body, but we don't know.
Thanks.
Mm-hmm.
Great, thank you, Michelle.
Yeah, thank you.
Our next speaker is Dr. Jose de la Torre. He's a professor at San Francisco State, and he'll be talking about metagenome and microbial population genomics.
Thanks, Athena. I had just gone to make myself a sandwich, but all right. So let's see if I can do this. So are you guys seeing this?
Yeah, you got it.
Okay. So I'm going to try to keep this very short today. Our lab works on a group of ammonia oxidizing archaea in hydrothermal ecosystems, and we focus very broadly on sort of all the aspects of biology of this, including physiology, evolutionary history, and population genomics more recently. These ammonia oxidizing archaea, or AOA, are ubiquitous in many environments. They're found in marine and freshwater environments where they can actually be up to 40% of the bacterial plankton. They're host associated, especially with marine invertebrates, especially cnidarians. They're found in soil, wastewater treatment plants, and also in hydrothermal systems. And in all cases, these organisms make a living by oxidizing ammonia aerobically to nitrite. And they can do so in environments where there's vanishingly small amounts of ammonia because they have a very, very high affinity for ammonia, their primary substrate, much higher than any other measured affinity for a chemical, energy substrate that's been found to date. And so our work has looked at these organisms in thermophilic environments, primarily initially in Yellowstone, but we've also been doing some work in Nevada, and also we had a 5-year project to look at the diversity of these organisms in hot springs. And part of the interest for these guys has been that they're an important player in sort of global nitrogen cycle. They regulate one of the key steps in the nitrogen cycle, in the aerobic nitrogen cycle. So from a point of sort of global geochemistry, they seem fairly important, especially in marine environments. So we have, through the efforts of a ton of students, some of whom are shown there at the bottom, we have cultivated a large number of these organisms from a diverse set of hot springs, some in North America, some in Asia. There are also 2 other groups that have cultivated strains from Iceland and Italy. And then we also have a ton of metagenomes and metagenome assembled genomes, this term that is horrific, but is being used to describe the sort of quilt-like assemblies of genomes that really represent more of a population than they represent an individual organism. But they're useful, as you'll see in a second. And so we've been looking at these, comparing them to try to get a better sense of the physiology of these organisms. But more recently, we've also been doing some more population genomics and also biogeography. So phylogenetically, when we look at these, The thermophiles form the deepest branch of these ammonia oxidizers. All the mesophiles, the ones that live at lower temperatures, seem to be derived. And we see a strong biogeographical signal. You can see it down here in the bottom right, that the ones from China and also Japan seem to cluster differently than the ones from North America. Interestingly, the European strains also group with this sort of Eurasian group. And this phylogenetic tree is actually a phylogenomic analysis of 122 concatenated archaeal marker proteins that is using a tool by folks at the University of Brisbane, which is great for classifying an organism. If you have a new bacterial or archaeal genome and you want to know if it's an existing species or not, you can put it through this toolkit, and it's really quite phenomenal. And it's sort of normalizing microbial taxonomy in the era of genomics. So this lineage that I'm showing you down here is exclusively thermophilic, but they're almost all almost exclusively terrestrial, and they've been missing from hydrothermal vent systems until just recently. And so last year, a group from Japan and Caltech published some metagenomes from an iron-rich hot spring in Japan that had the first thermophilic marine organism associated with this clade, and it looks like it is related to 16S sequences that had at this point been unaffiliated with any sort of physiology, that were found in a shallow hydrothermal system in Papua New Guinea by folks at USC and the University of Chicago. So when we look at these thermophilic genomes and compare them to each other, what we find is that the average nucleotide identity across the entire genome within a region is extremely high. So it's usually around 99 to 100% identical across the entire genome. So this is really quite remarkable and consistent. And I should say that these genomes are small. They're only about 1.5 million base pairs, so easy to work with. And what we see is sort of reminiscent of that phylogenetic tree is that as you go further, you have very different, much lower average nucleotide identity between these different clades, to the point where we can essentially say that the North American group appears to represent a separate genus from the others. It's still debated whether the European and the Chinese lineages are different species or different genuses of the same organism. To some extent, these things are semantics, and so in this plot, what we've done is we've essentially plotted on a logarithmic scale the shared genes, which we call the core genome, and then all of the sort of unique or auxiliary genes that are in each genome. And what this allows us to say is that there's very little variation in sort of the core genes that are conserved across these organisms. And for us, this is interesting because it helps us get at sort of what the energetic engine of these organisms might be, since a lot of the physiology and biochemical pathways are poorly understood. But in those genes, in sort of the open circles above, those are the genes that are really driving adaptation to specific niche environments. And those are the ones that are interesting in terms of sort of phylogeography and things like that. And so a fair amount of work has been focused on those. When we align the whole genomes of these things, and so these are all 1.6 or 1.5 million base pairs of the genomes. What you can see is that the genomes are remarkably synthetic. In other words, the gene order is extremely conserved. The regions where you have those white gaps, it's hard not having a pointer. The regions where you have those white gaps, those are regions of differences between the genomes. And what we found is that in some places you have a cassette of genes that are swapped at a specific location, and we tend to refer to those as flexible replacement islands. And what we find is that a lot of those genes, similar to what has been found in marine bacteria, these are genes cassettes that are driving things like glycosylation or outer membrane proteins, and those are essentially changing what the outside of the cell looks like. And it's been argued that that is a way of evading viral predation. We heard how viruses dominate about 10 to 1 the number of cells in an environment. So there's a very strong selection to be able to change the external surface of the microorganisms. Some of the larger regions, though, are mobile genetic elements, and those are present in these genomes at a much higher density than we've observed previously. And so that's been become a focus of work in the lab by a number of students, and some of this, so some of these are known as ICEs, so integrative conjugative elements. The most famous of these ICEs is the F plasmid in E. coli. And what these things do is that they integrate into the host genome, sort of like a virus, and then to replicate, they excise and replicate as a plasmid, and then are transferred to naive recipient cells using, at least in bacteria, type 4 secretion systems. So these are pili that extend between the cells that allow the transfer of single-stranded DNA from one cell to the other. And this is a manner in which these things replicate. However, in E. coli, we know of lots of cases where these things are permanently fixed in the genome, for example, if they lose the integrase. And we think that that's what's happening in these organisms, in these archaea. We can also look in metagenomes, and we've looked in many metagenomes that are available on NCBI or at the Joint Genome Institute IMG website. And do recruitments to try to figure out the abundance of these ammonia oxidizing archaea in those hot spring metagenomes. And what we find is that they're not present in most of the genomes that are publicly available. But when they are, they're actually quite significant. And it seems to be always the case that they're in near circumneutral hot springs, so pH between 7 and 9, and at temperatures above 65 degrees. And in particular, we find them most commonly in some of the hot springs that we were sampling in China. But the nice thing about this is that these metagenomes represent population level genetic information that we can then start looking at to look at variation in the genome. So in this case here, what we have is we have 3 of our genome aligned at the top of the panel here. And what we've done is we've recruited individual reads from the metagenome to the genome that's on top, that DRC1 genome, and plotted it by its position and its percent identity. The percent identity is given on the right side of this central figure. And what we can see is that there's a lot of these organisms present, but we can also see large gaps in the recruitment that correspond to some of these genomic islands. And the one on the far right, for example, where there's a significant amount of reads down around 70 and 75%, that's actually a massive CRISPR-Cas operon that clearly is going to be driven by sort of the local environment of that particular microbe or that genome. And so it's not surprising that that might be more variable. And at the bottom, in sort of light blue, what we have is the coverage. And you can see that on average in this metagenome, when we select reads that are only 98% identical or more, which corresponds to about species level divergence, then on average, the coverage is about 40 across the board. But we can see some regions where there's no coverage at all, like that CRISPR system at the end. But there's other regions where there's a much lower coverage. And that indicates that in some of those areas, that those regions of the genome might actually only be represented by a small portion of the microbial community or of the population in that hot spring. What we're trying to do now is select only those reads at 98% identity or above, and look scanning across the genome to map SNPs and see which areas and specifically what genes in the genome seem to be the most variable, and try to see if there's a pattern there that we can look at. When we do the same thing, oh, and I should have said that on the histogram on the right represents the percent of the matching reads, and you can see that the 100% range in this slide is very high, and that represents the majority. But there's a lot of reads that are 95% identical or lower, and those represent close relatives that are probably the same genus but a different species. And unfortunately, we never get those in culture. So we're trying to devise strategies to use these metagenomes to assemble only those genomes that are more distantly related, but that's still a work in progress. Okay, and so here you see a clear example. This is our Yellowstone genome recruiting reads from a hot spring in Nevada, and you can see big gaps in the recruitments that correspond to these genomic regions that are much more variable. And you can see on the histogram that in this spring in particular, there's not a very close relative. The closest relative is around 95% identical, which indicates that it's probably the same genus but a different species. And so here, in the genome of that Yellowstone organism, what we found is that one of these mobile genetic elements is actually still mobilized as a circle. And this was work that was started by Virginia Russell when she was still in my lab before she went off to do her PhD at Berkeley. And she is on this call, so if anyone wants to ask a question, Virginia can answer it. And what she and a new student, Rachna Panu, have been working on is characterizing this mobile genetic element, this ICE that actually is as a plasmid most of the time. And we're looking at trying to determine what the mechanism of propagation of this genetic element is, but also to see if we can use it as a vector for heterologous expression, since none of these organisms, whether they be from marine, soil, or hot springs, have any tractable genetic systems at this point. And then I'll just conclude that we see some very interesting patterns. For example, there's one mobile genetic element that seems to be permanently integrated into the genome that is almost entirely conserved in all the Chinese, Japanese, and European strains that we see. So these things have been in the genomes for a very, very long time. And they may be driving why these genomes are so syntenic. And the rationale for that is simply that these organisms may actually be transferring large portions of the genome when they conjugate, similar to the F plasmid in E. coli when it becomes permanently integrated in the genome. So instead of just transferring just the plasmid itself, it may be transferring a large portion of the chromosome as well, which would keep the gene order fixed in this population. All right, so I'll stop there. Lots of people have worked on this. And so we've been really thankful for having a lot of collaborators around the world. And I will stop there and take questions if there are any.
Okay, really quick question. Virginia asked, are the European AOAs from iron-rich hot spring as well?
No, the European AOAs are not from iron-rich springs.
And Aaron, you have a quick question?
Yeah, I do. Hi, thank you. So I know you probably have difficulty dating these different lineages of archaea. I'm just curious if you notice any kind of pattern in the formation of different different, geothermal areas where these archaea are found, and sort of the resolution on the phylogenetic tree.
We have not been able to do any sort of dating to put this into a sort of a timeline context. The plumbing on these systems is very, very dynamic.
Mm-hmm.
Obviously, we can know when continents spread apart, but all of the data suggests that there's still gene flow happening between the Chinese and the European strains, for example. How that works, nobody knows.
All right, thank you.
Great, thank you so much, Jose. Okay, our next speaker is Dr. Roxanne Banker. She is a postdoc at the Cal Academy, and she's going to talk about oysters.
Hey there. So if anyone tuned in for my talk last week, this will be a lot of similar stuff. But here we go. Can you guys, let's see, I always got to figure this out. I also think I did manage to turn off my closed captioning for anyone that was there for that snafu. Anyway, so my work on my dissertation, I did a brief postdoc also at UC Davis focused on oyster microbiomes, specifically with regards to how the microbiome might affect shell formation and the formation of different shell features. And so what I'll be talking about today is based on a brief postdoc I did there. So larval to juvenile oyster shell development, a multi-omics approach. So the objective of this work was to expose oysters to conditions that affect shell formation. Specifically, we were thinking of low pH in the context of ocean acidification. And then we wanted to assess, do these different conditions affect the shell? How does the microbiome respond to changing environmental conditions? How does gene expression change in response to changing environment? And then how might these things interact to affect the ability of the animal to produce its shell material? And one thing that I thought was really cool that Javier said in his talk was that his insects were empowered by microbiomes. And it's a really good way of encapsulating the way I've thought about oysters and marine tax a lot. And I also think that the microbiome so far has been not thought of a lot in the context of marine skeleton formation. So that's sort of how I'm thinking about this. So just a little bit of background. I'm sure most people here are pretty familiar, but ocean acidification is the result of us putting CO2 in the atmosphere. CO2 dissolves in the seawater and increases acidity. This reduces the carbonate saturation state in seawater, and both of these things decrease the ability for marine calcifiers to produce their shells normally. So it can either dissolve existing shell material or create barriers to producing new shell. And oysters are both ecologically and commercially important species. Their filter feeding behavior really helps to improve water clarity, especially in systems that have been subjected to anthropogenic eutrophication. And then they're also very commercially important. So they're eaten all over the world, and they're a pretty important food product. And ocean acidification has already been a significant challenge for the aquaculture industry. They've experienced mass mortality events and had to come up with other ways of making sure that their product can reach market stage. And the early life stages are really sensitive to ocean acidification. One other interesting thing about oysters that informs some of the questions that I want to answer is that oysters actually produce two forms of calcium carbonate in their shell, aragonite and calcite. So they have the same chemical composition, but are arranged atomically differently, so they have different material properties. Aragonite tends to be better at like flexing and bending, whereas calcite is just generally more stable and more resistant to acidification. So just a little bit about oyster development. Oysters begin life as a planktonic larvae, and they spend about, I don't know, depending on the species and the temperature, 3 or 4 weeks swimming around in the water column before they lose their ability to swim, and they settle to the seafloor, find a hard substrate, and cement themselves there permanently. And so here's a picture with days on the x-axis and shell size on the y-axis, and just a few little pictures of the different stages. And so you might start as an embryo at day 0. After about 2 days, you reach the D stage, which the shell is shaped like a D. And then the oyster quickly grows rapidly until it reaches this eyed pedovelliger stage. And that's the point when it loses its ability to swim and settles and will then grow into a juvenile or adult. So the way we set up this experiment is we wanted to grow oysters from that larval stage to their settlement stage under various conditions to see how that affected shell formation and other things like that. And so we grew oysters in normal pH conditions, acidified pH conditions as an analog for ocean acidification. And a third treatment, we had normal pH conditions, but we added a compound, sodium molybdate, which is known to inhibit bacterial sulfate reduction. And the reason we were interested in inhibiting that specific metabolic group was because they seem to be very important for carbon cycling, and they seem to be important for carbonate precipitation in microbial map systems. So that was another sort of exploratory treatment. And the way we chose the Pacific oyster for this work, because it's a very fast-growing common aquaculture product, so it's relevant to stakeholders. So we actually spawned our own oysters for this experiment, which was new and kind of cool for me. Here are the big holding tanks in the background. And then the process of spawning is we had a number of brood stock, and we went through and checked for gametes. When we found enough males and females that had gametes, we combined those and then grew up the embryos in one of these really big tanks. The larvae were allowed to reach the D-hinge stage, which takes about 2 days again, before they're transferred to their buckets, which you can see back here, which is how they were cultured for the remainder of the experiment. And at this point, the group culture was what was sampled for our larval time point, and I'll talk about that more in a moment. So again, the experimental design was we grew up the larvae in this large pool population, and then once they reached that D-hinge stage, which is also, I forgot to say, when the shell totally envelops soft tissues, and they're a little bit more durable to perturbation. So then we split that pooled larvae approximately equally across the 3 treatments. And for each treatment, we had 3 replicate buckets. This is how it actually looks. So it's not quite as neat and tidy as that. But the way that pH was controlled is that these are pH monitors. And so there's a probe running into the acidified tanks here. And when that probe registers the pH that's too high, that'll activate a current here, which opens a valve on these CO2 tanks, which then runs CO2 back into the bucket to drive pH down. All buckets also had an airline to keep them oxygenated, and they were fed a diet of algae every day. We monitored sea temperature, dissolved oxygen, salinity, and pH. And then because these are static cultures, so there's not fresh water running through the system, I had to manually change those. And during each water change, I also took additional water samples for pH to be analyzed on a spectrophotometer to get a more precise value for pH over the course of the experiment. And then the way we sampled, so from those big spawn cones where we cultured the larvae, we decanted some water, poured it onto a really fine sieve. So this is actually a sieve that I think has about, I'd have to check, but about a 35 micron size. And then these little pieces of sand are actually larval oysters. So we isolated them onto a sieve, scooped some out, and froze them in microcentrifuge tubes. 3 samples to have RNA extracted for a transcriptome, 3 samples for DNA extraction for the microbiome. And then we use approximately the same procedure to sample juveniles after they'd settled in the buckets. So I had to scrape those off with a spatula, and we sampled those 12 days post-settlement. So I destructively sampled buckets 1 and 2 from each treatment. And then the last bucket was destructively sampled 25 days post-settlement. So it was bucket 3 for each treatment. And I'll just show just a little bit of data. So this is a principal coordinates analysis plot based on Bray-Curtis distances. And if you have any questions about the bioinformatics I used, I did most of it in R and the Data2 pipeline, and then fed the resulting information into PhyloSeq. So these are adjusted sequence counts using the variant stabilizing transformation in DSeq, and then Bray-Curtis is run on that. But it's cool because the control samples here are shown in brown, and they tend to cluster very well together. And even more than that, they actually cluster based on the timing of their collection. So 12 days post-settlement has a distinct community from those that were sampled 25 days post-settlement. And that pattern is also mirrored in the other treatment groups. So SM, sodium molybdate added. Interestingly, the 25-day molybdate and 25-day control samples seem to have a similar community composition. And I'll have to dig into the why of that later. This is an ongoing project. And then you can also see a similar pattern in the low pH or OA samples here. So next steps are figure out what's driving the differences between treatment groups. So how are the environmental conditions affecting the differences between treatment groups? What are the taxa that are different between time points? And then also I need to start looking at that shell data. So I'll be measuring the difference between the larval shell, which you can see clearly as compared to the juvenile shell length, and then assess whether there are any correlations between certain taxa, community characteristics, and the state of the shells in this treatment group. Right, I think that's about it. So any questions? Oh, I should stop sharing.
Thanks, Roxanne. Any questions? All right. We're running a little over, so
yeah. Great. Well, if anyone has questions, please email me. Thanks.
Great. And our next speaker is Dylan Chivian. He is a computational biologist at the Lawrence Berkeley Lab. Take over, Dylan.
Great, thank you. So let's see if I can do this properly. Trying to share, but it doesn't seem to be doing it. Maybe I can do it this way. Can you tell me what you're seeing?
We see a PowerPoint. So just click
You do.
send from here.
Okay, great. And I'm hoping it goes full screen now.
It says loading. Okay, you got it. Perfect.
Okay, fantastic. So I'll try and be very quick. In essence, this is an advertisement for a resource we've developed and are continuing to develop for everyone. We are funded by the Department of Energy, and a lot of the work is environmental microbes and not human associated, because that's NIH's domain. But frankly, the tools that we're developing can be used for anything. So no matter what your area of research, we encourage you to come check us out. So I really don't have a lot of time. So instead, like I said, it's an advertisement, and I would like to point you to some upcoming webinars where we actually go into the details of how to use the tools in particular areas. If you want to see what the upcoming webinars are. Can you see my cursor, by the way? Does this work? Maybe not.
Yes.
So we have, okay, good. So we have this URL here with our list of our upcoming webinars, and you can watch old ones that have been recorded as well. The next one is on metabolic modeling of microbiomes. I have one that I'm giving later this month on phylogenomics, and then I'm doing one next month on microbiome fractionation and what the tooling is there. So KBase is the DOE Systems Biology Knowledge Base, and what that is it's sort of, we have kind of a twofold mission. The first is to sort of provide as much as possible a one-stop shopping and compute resource for people to do their bioinformatics and modeling of microbiome. So we bring in all public data from NCBI and so on. We integrate it. We allow you to bring in your own sequence reads or assembled genomes or RNA-seq and so on, and analyze it against public data or against your own data. You can work with your collaborators, share these things. We use the Jupyter Notebook as our framework, and so these are computable notebooks, and we build interface to sort of simplify things. And so we take a lot of existing bioinformatics tools that traditionally have been run on the command line, and we put a GUI to it, and it becomes more like an app in how you use it. And then, so that's our sort of one aspect of our mission and effort. The other is to take all of this data now that it's all together and link it by homology and terms and natural language processing and so on. And we're working towards this a knowledge engine, relation engine based approach where you can infer information and models from the relationships between those data. So ultimately, we want to get to predictive models. So you could imagine trying to have, if you had conditions and lineage information, you could infer what function is, or if you had lineage and function, you could infer conditions, or conditions and function, infer lineage, etc. So being able to go from one to the next, that's a predictive model. Our overall picture of the framework is being able to take things from the wild, do direct bioinformatics on that data, but also maybe you have isolates or ways of building synthetic communities, and you want to be able to measure their response to perturbation or conditions, and put that all together and build out models, do models of interaction, and so on. So that's the framework we're trying to offer. The thing I've been really interested by lately is, so we offer a lot of tooling for genome and metagenome assembly and metagenome assembled genomes, or MAG, and those are genome-like entities, quilts as it were. And those are very valuable because then you can link function to lineage But additionally, you may have unbinned contigs that at least you can still get some notion of operon and synteny and gene co-occurrence. And those are useful as well. And then there's the reads that don't assemble, and those may actually carry much of the functional complement of that microbiome. And so you want to be able to analyze sort of each of these three fractions for lineage and functional complement. And so that's sort of the framework we're heading towards. You may start out with just raw reads, and you want to go through and get a sense of which lineages are there. You want to do read hygiene, so we have tooling for that. We have several tools for lineage assessment on reads, shotgun metagenomic reads in particular. And so you can get pictures of what the lineage complement is, and there's these pretty coronal plots you can get to. We have both isolate and metagenome assemblers. We have several of them, and you can compare the results of them. You can compare the results of single library assembly versus co-assembly and so on, get histograms of contig lengths and so on, and pick which ones seem to be performing best. You can then take a metagenome assembly, or you could upload one if you didn't do it on system, and use various binning tools, which is a way of associating the contigs from a metagenome assembly into a putative lineage cohort. And then you can use this tool, Check'em, to assess the completeness and contamination of that quilt's genome. Population genome is another term. And then you could extract the high quality or the high quality and medium quality mags from that. So I don't have a lot of time, and I just want to mention that we additionally have quite a bit of phylogenomics and functional assignment to these genes and genomes, but it's too much to go into here. So again, on April 22nd, I'm giving a webinar on all of this. And I think I'm giving another quick blurb at later one of these social hours. But so we can build gene trees from your mags. I'm currently working on expanding our support for viral phylogenomics, since it's kind of important. And then we can take those species trees and scan their genes for function and determine which lineages are carrying which function. And this is also upcoming, is the ability to look at the genomic context of those genes against either gene trees or species trees. So I want to thank the KBase team. We're a multi-institutional effort between Lawrence Berkeley Lab, Argonne National Lab, Oak Ridge National Lab, Brookhaven, and Cold Spring Harbor. And so this is some of the team, and here's some more of the team in the Bay Area. And I want to thank you.